Give me a sec. I'm gonna share my stuff. Put it here. Third one on. Okay. Everything that I need to set up. Okay, put it there. Hi，新来的观众，安安你好，你好，你好。那今天早上呢，我想要来练习一下。我明天要上台去报一个科学的论文，那我今天想要先练习一下要怎么报这样。This morning I'm going to practice delivering a speech on my scientific report, as I'm going to make an official one tomorrow. So yeah, here's it. So hi everyone, good morning. My name is Laurent, and today I'm going to talk about the interaction between tubular epithelial cells and macrophage in the development of tubular interstitial inflammation, and which caused by HF1 alpha and microRNA 23A. So first of all, let's talk a little bit about the renal system. So here is a schematic graph I draw for the kidney, and our blood will enter this blue renal artery and enter the renal medulla, which consists of thousands of millions of small nephrons, which is the basic functional unit of the kidney. Here, in the nephron, our blood will be filtrated by glomerulus, proximal, and then two distal tubule. The reusable substance just said some protein, amino acid, and ions will be reabsorbed, and the rest of them are considered waste and be propelled into the urine, go to the bladder, and secrete out our body. When damage strike our kidney, such as drug or infectious or genetic or many other reasons, if a damage strike our kidney, the first thing that will happen is tubular interstitial nephritis induction, or you can also say tubular interstitial inflammation. They are the same thing. So in short, TIN or TI. So this is a phenomenon which characterized by the infiltration of macrophage in the kidney interstitium, which is the area that located between these tubules, the interstitium. So the immediate effect of the strike is the acute kidney injury, and if not healed and stay for a long period of time, this will lead to chronic kidney disease and eventually kidney failure. So one key message here is that it's very important to stop or to block or heal the kidney damage at an early time point before it's going to the point of no return. Okay, now I'm gonna talk a little bit about our first main character, tubular epithelial cell, or in short TEC. So it's the basic functional cell types that in our kidney tissue, it absorbs the reusable substance in the primary urine like glucose and amino acid, etc., and you regulate the balance of water and electrolyte. Tubular epithelial cell, though, however, is very weak. It's vulnerable to injuries such as hypoxic, ischemic, obstructive, and metabolic insults. And due to the reason, it was considered as the poorest victim in the progression of kidney disease. Okay, as I just say, it was. Recent years, more and more evidence have demonstrated that rather than just being merely a victim, tubular epithelial cells actually participate in the progression of kidney disease. Okay, for instance, in the previous report of this the same author, he demonstrated that TECs, when overloaded with albumin, will induce TIN through the TEC macrophage interaction mechanism. So, the next, having done with TEC, our next character is HIF1 alpha. This is a master transcriptional regulator of our adaptive response to hypoxia and is encoded by HIF1A, which is hypoxia-induced factor 1 subunit alpha, the full name of it. Okay, so under hypoxia, HIF1 will activate many genes involved in metabolism, angiogenesis, apoptosis, and many other genes to facilitate the oxygen delivery to hypoxic tissue. However, though, there is reported that the inappropriate activity of TEC released HIF will actually promote interstitial fibrosis. Furthermore, there is a crosstalk between HIF1 and alpha B, so which link that the hypoxia to the inflammation. Okay, so our next character in the game is microRNA or miRNA in short. So it's a non-coding RNA. Its single strand is about 20 to 21 nucleotides, and it regulates messenger RNA post-transcriptionally. These miRNA are synthesized within the nucleus. They are here the premature miRNA are processed by Drosser and Dicer, then propelled out of the nucleus, go into cytoplasm. The mature miRNA will then form complex with Argonaut, forming RNA-inducing silence complex, the RISC complex. RISC complex, along with its companion RNA, will then target messenger RNA. It will either repress the translation process or will drive the degradation of the target messenger RNA. Okay, the miRNA that is in interest in our study here is miR23A. In a previous paper, it's reported that the transfer of miR23A via exosome can regulate the immune consequence, and which is why we're interested in it. Okay, the last but not least, our final characters in this story, exosome or extracellular vesicle. Exosome is synthesized, is produced by the endosomal compartment of the cell, who first of all is in the form of endosome. Then this endosome, if the membrane of it is burned inward, it will form intraluminal vesicle, which contain many substances inside it. So the whole thing is called multivesicular body, MVP, and the vesicle inside this multivesicular body is called ILV, intraluminal vesicle. When MVB fused to the membrane of the cells, ILV will be propelled out of the cell in the form of exosome and be received by the recipient cells. And these exosome can carry a variety of substance, for example, miRNA, RNA, DNA, proteins, lipid, and more. And this is one of the very important mechanisms for cell to to, how do I say that, to communicate with adjacent cells. For example, also in the same the previous report of the same author, they demonstrate that when TEC is overloaded with albumin, it will respond by release exosomal CCL2, which will receive by the macrophage, recruit them to the damage site and induce TIN. Okay, so here is it. The hypothesis that the author is making in this study is that HIF1 expressed in the TEC under hypoxia will regulate the macrophage function through an exosomal manner. Okay, and so the motivation of this study is that if we have an insight into the mechanism underlying this hypoxia-induced TIN, we can provide a new therapeutics approach for early blockade of the progression of renal disease. 皮蛋早安啊！哦，今天早班，今天7点多，你不是7点上班吗？这么一大早就来学英文，辛苦了。Okay, so before we go into the details in the result, because there are 9 figures and there's a lot of things that are kind of similar, so you guys might get confused and I might get confused too. So yeah, before going to the detail in the result, we just jump to the conclusion and see what is the findings of these studies. Okay, so first of all, here's a healthy TEC. Under hypoxia, HIF1 alpha will be the expression will increase in the TEC, and the result is that TEC will respond by releasing Mi23A enriched exosome. These exosomes are received by macrophage. As a result, inside the macrophage, Mi23A will inhibit A20, which is a negative regulator of NF-kappa B. So as a negative regulator, NF-kappa B is shut down, NF-kappa will increase, and the macrophage will be reprogrammed into a pro-inflammation state. Finally, the final consequence of this whole process is the development of TI, tubular interstitial inflammation. Okay. Now, let's talk about the result. Okay, let me sip a little bit water. Oh my goodness, it's so long. Okay. So first of all, in this study, the author want to examine the injury model and to confirm the previous showing that there is an association between TI and HIF in hypoxic kidney. Okay, and after that, they will confirm the existence of Mi23A enriched exosome in these hypoxic TECs. After finding out that, okay, so all the fundamental all the fundamental informations is gathered. Next up, they will try to test out to investigate whether there's an association between exosome Mi23A and HIF1 in hypoxic TEC, and in association with the tubular interstitial inflammation in the in vitro model and in vivo model. So first of all, let us see the association between TI and HIF in hypoxic kidney. They use 2 models to examine this phenomenon, issue. The first model is ischemia reperfusion. It's called also IR model. So ischemia is the insufficient of blood, and reperfusion is the coming back of the blood. And the mouse that are used in this model is a 6 to 8 week old male C57BL-6 mice. In this mouse, both of the renal pedicles are clamped for 30 minutes. So in this state, because it's clamped, the blood vessel along with many other vessels is not working because of the lack of blood. And so this is what we call the ischemia state. After 30 minutes, it's released, and this the reperfusion state. This will cause a damage on our kidney tissue because in ischemia stage, a lot of RS will be produced, which will drive the damage of DNA, protein, and degradation of membrane lipid, which eventually lead to inflammation. However, though, because it's like a reversal model, because like we see that it's only clamped for 30 minutes. After that, it's released. So in this model, the kidney will eventually it will be repaired. Another model though is an irreversible model. It's called unilateral urethral obstruction model. In short, UUO. This also uses 6 to 8 week old male C567B-6 mice. And in this model, the right ureter, so only the right one, this is called why it's called unilateral. Only the right ureter of each mice is dilated. So this is how they do it. So they put a ligature, they obstruct it inside the kidney. It's called kidney pelvis. So yeah, as you can see here, this thing, when this thing stuck, so this thing will be stuck inside the kidney, and all the blood, the urine will cannot be, and the urine will not be able to flow through this ureter. As a result, the function of kidney, well, basically it lost mostly of its function at this point, and this is an irreversible damage. Okay, so using these two models, there are several things that we want to know about it. So first of all, we will examine whether the injury model can be worked. Okay, so we use kidney function, and we examine the kidney function and its histology using creatinine and blood urea nitrogen, BUN. These two are the products, the metabolic products that are actually waste and should be filtered out of our blood. So if our kidney function are actually going down, these the level of these molecules will goes up. Okay, histologically, plastin, which is used to stand for polysaccharide such as glycogen, glycolipid, and these are especially abundance in the kidney and liver, those type of cells. So we can use this to stand for this cell and to see the morphology change of it. Other than that, we also stand for F4-AD macrophage to see whether there is an infiltration of macrophage, which indicate the induction of tuber-interstitial inflammation. Okay. Finally, if we know that okay, this model is usable, we also examine it in the inflammatory markers to see that whether these markers are increased. And finally, we also will see that whether HIF will be induced corresponding with the time course of these inflammatory markers to see whether there's a link between the TI formation and HIF1 induction. Okay. So first of all, is that using the IR model, because okay, so here they don't use the UUO model because apparently, as you can see, that UUO model is totally is the kidney function is totally dead. Okay, so we don't have to examine this anymore because yeah, you just can't, because in this model they will collect the serum creatinine and blood urea nitrogen, but there is no yeah. Okay, so as you can see that in figure A, the y-axis is the level of these molecules, the x-axis is the samples, okay, and we have sham day 1, day 3, and 7. And apparently, it's pretty obvious that there is a significant increase in the level of these two molecules, the serum creatinine and BUN, at day 1, and especially at day 1 and day 3. After that, it coming to a kind of like a decrease at day 7 compared to day 1 and day 3. That is because in this IR reverse model, at about day 7, the kidney will start repair itself. So the function, the kidney, and it will gain back a function. Okay. And next up in histology, for the IR model, we can see that in the past tense, so compared to the sham at day 1 and day 3, we can see that there is a structure, the morphology destruction is going on in the kidney, and it slowly become a little bit recover at day 7. The same things go with the infiltration of macrophage. There's an increase of macrophage infiltration at day 1 and day 3. Okay, for the UUO model, we also see a very similar corresponding results, which show a rupture in the morphology of the kidney tissue at day, but a little bit more vigorously because it's an irreversible model. So even at day 7, there's the destruction keeps going on, and the infiltration of macrophage is even more intensive. Okay, so now let's move on to the molecular level. In the IR model, okay, so in the molecular level, we will the inflammatory markers that we investigate is MCP1, TNF alpha, interleukin 1 beta, and phospho P65. Okay, so in figure D, which is a qPCR result, we can see that compared from different sample, compared to sham day 1 and day 3 samples, there's a significant increase in these inflammatory markers, which preceding a decrease at day 7 following the recovery of the kidney tissue. In Western blot, the same things, the same pattern goes for the phospho P65, as there's an increase of them at day 1 and day 3 in the quantized group. In the quantized figure, we can also confirm this phenomenon. Okay, so okay, yep, that's okay. So the key message here, okay, the key message here is that these inflammatory markers, observed at day 1 and day 3. Okay, and in the UUO model, the similar pattern is also seen, however, with a more intensive response, as we can see that the inflammatory markers is the increase of the inflammatory markers is even more strong. Wait, is it? Okay, never mind it. So also an increase in these inflammatory markers, as well as phospho P65. Okay, so so we know that the time point where these inflammatory markers increase, actually they indicate the above experiment indicate, give us a message that when will TI will occur in terms of histology and molecular. Yo, morning, man. morning. Dude, are you still not going to work? I'm practicing delivering a scientific report now. Okay, I keep continuing. You just keep talking to me. Gotta go to work now. Bye. Okay, dude. See ya, bitch. Okay, so now the key message that we learned from these previous examination is that in terms of histology and molecular level, there's TI, the formation of TI is induced at about day 1 and day 3. So we want to know whether HIF1 alpha, our main character, are induced at about the same time. So if they are, then in this proof that there is a possible association between these two, between HIF1 alpha and TI induction. And so we can see that in RT-qPCR there's an increase in TI at day 1 significantly. In terms of protein using Western blot, we can see that there's an increase of TI at day 1 and day 3 after the IR kidney injury. Here's a contrast graph of that. And using immunohistochemistry staining, we can see that the yellow indicate excuse me. Okay. The yellow or brownish color indicate the existence of HIF, indicate the presence of HIF1 alpha, and we can see that they are present at day 1 and day 3, which receding a decrease at day 7. Using UUO model, we also see a very similar pattern, of which HIF1 alpha messenger RNA increase at day 1, day 3. The protein increase also at day 1, day 3, and immunohistochemically they present in day 1 and day 3, preceding decrease at day 7. So from here we can see that the inflammatory markers, these MCP1, interleukin 1 beta, NF kava, and phospho P65 expression in both kidney injury model correspond to the time course of the induction of HIF1 alpha, which indicate that there's a link between TI and HIF1 alpha. Morning. I'm practicing. I'm going to give an official one tomorrow, so yeah. Okay, so now that we know there is some sort of association between TI and HIF1 alpha in these hypoxic kidney, the next thing we want to know is that whether there is actually Mi23A enriched exosome in these hypoxic TECs. Thank you. Okay, so in this section, first of all, we will confirm the existence, and next thing, we want to examine whether these Mi23A functions is related to the development of TI. Okay. So here, the author use ZW nanoparticle tracker analysis to characterize the size of the kidney exosome, and also using Western blot, ALIX CD63 and CD9, which is exosome markers. They confirm the existence of exosome of these kidney exosome. at day 1, day 3, and day 7 after the injury. Yeah, no, it's also appear in the sham too. Yeah. Okay. So after confirming that the exosome actually exists, the next thing they want to know is whether there is MIR23A inside this exosome. So first of all, in figure C and D in the IR injury kidney and usual kidney model, the kidney lysae are collected and the exosome are extracted using differential centrifugation. Then they use these exosomes are addressed to RT-qPCR to see that whether there is MIR23A inside it. And as we can see that there is a significant increase of MIR23A at day 1 and day 3 after the injury in both models. To be more specific, the author extract the tubules from these injured kidney and extract the exosome from and purify exosome from this tubule and do the same experiment, and also confirm that there is significant increase in the MIR23A. So here, oh yeah, I forgot to mention that in figure E and F, E is at day 1 after the injury and U is day 3 after the injury, accordingly to because this can, as a result in figure C and D, there's an increase in day 1 in the IR kidney, and there a significant increase in day 3 at the usual kidney. And so, okay, let's moving on. Okay, so we can see that similar to the inflammatory markers and phospho P65, MIR23A enriched exosome, its expression is also corresponding to the time point of HIF1 alpha induction, which is at day 1 and day 3 after the injury. So this also this suggesting that there's a link between these MIR23A enriched exosome and HIF1 alpha. So now we learned about the existence inside our hypoxic TEC. Now we want to know why is the function. It's related to TI development. Here the author use gain of loss of function experiments, so they inhibit MIR23A, and as a result, we can see that in terms of function, kidney renal function, we can see that inhibition of MIR23A can bring down the level of serum creatinine and BUN, which indicate that inhibition of MIR23A will ameliorate the function of, to improve the function of our kidney. Histologically, inhibition of MIR23A also improves the morphology of the damaged kidney and decrease the filtration of macrophages. In terms of molecular markers and phospho P65, we can also see that there is a decrease in these inflammatory markers when MIR23A is inhibited, as well as phospho P65. Okay. So now, so far we confirmed that there's association between TI and hypoxia 1 alpha, and we confirmed that MIR23A enrichment exosome exists in hypoxic TEC, and MIR23A itself function is related to TI development. Now let's move to the kind of like the main story, okay, the main quest in these stories, okay. And it says to investigate the association between these axomitoria and HF alpha in the development of TI. First of all, in the in vitro model. Okay, so in this section, first of all, we will confirm the link between these axomitoria and HIF1 alpha. Then we will narrow down to the details of how the axomitoria and the HIF1 alpha interact in hypoxia. The in vitro model that the author using here is a mouse TEC cell lines, and these TEC were exposed to 1% of oxygen for 24 or 48 hours. And here the author tests out that in 24 or 48 hours, which one is might be a better time point for the experiment. So they measure the mitochondria level in these two conditions, and as a result we can see that okay, so in this is the qPCR result. We can see in 24 hours first, compared with the control exosome, the hypoxic in the compared to the control, the normoxia group. In the hypoxic group, mitochondria level in the 24 hours there is a very slightly increase, and it's not significant. However though, in 48 hours the increase of mitochondria in TEC in the hypoxia group compared to the control group is significant. Okay. Also, to specify these issues, the authors also examine mitochondria level in exosome, and as we can see that there's even a more significant change in the level of mitochondria under hypoxic condition in the 48-hour group. Damn, this 9 figures is so long. Gonna sip a little bit more water. Keep hydrated. My mouth needs to rest. Ouch. Okay. Okay, where are we going here? Oh, okay. Okay, so to investigate whether HF1 alpha is related to the expression of mitochondria, the author want to perform get a loss of function experiment. So they use siRNA as a knockdown how do you say that method. So figure C is just that they using Western blot, they confirm that the SIHF1A actually work. The treatment of this siRNA will decrease the expression of HIF1 alpha. So then they will use this SIHF1 RNA to treat TECs under normoxia or under hypoxic condition. And as a result, first of all, we can see that in terms of mitochondria in TEC using qPCR, is that under normoxia, the treatment of the knockdown of HIF1 does not significantly affect the expression of mitochondria. However, though, in hypoxic condition, the knockdown of HIF1 alpha, the knockdown of HIF1 alpha create a very significant change, a decrease in the mitochondria. There are similar things also observed in the expression level of mitochondria in exosome, as we can see that if the exosome if HIF1 alpha is inhibited in the hypoxic TECs, mitochondria level is significantly decreased. So the other also use chromatin immunoprecipitation to check that whether there is a direct interaction between HIF1 alpha and mitochondria. So in the chromatin in the chip, okay, move back. Damn, I should get breakfast before this. It's just so dizzy, man. Yeah, it's actually get dizzy because there's so many stuff going on. Okay. Hold up yourself, Laurent. Okay. So in the in chip assay, we can also confirm that HIF1 and alpha will occupy to the promoter of mitochondria. Okay. So the short summary here is that HIF1 alpha will induce exomitochondria expression through the direct interaction in the hypoxic TECs. I'm gonna sip another water. Why so long? Why the hell I picked this paper? It's 9 figures, god damn it. Why am I so stupid? Take a little bit rest. Okay. Okay, let's get back on track. Okay. So now that we know there's a link between these exomitochondria and HIF1 alpha, now we want to know what's the mechanism underlying this interaction. Okay. So here the authors will first of all, in the bigger scale, they will investigate the interaction between TEC derived exosome and macrophage, and in the smaller, the more detailed scale, they will try to investigate the interaction between these exomitochondria and macrophage. In the bigger scale, first of all, the author perform a transfer study. So here's the upper chamber is filled with TEC, and the oxygen here can be regulated, so the author can decide whether they want to perform a hypoxia experiment. And the upper layer is that, and the down layer is filled with macrophage, and there's a permeable membrane that allows the exosome to derive from TEC to go through it and be absorbed by macrophage. Exosomes are stand green, as you can see here, and the macrophage here is a mice macrophage, which is called RAW264.7, and compare with the control group, we can see that in the hypoxia group, these exosomes, more of them are internalized into macrophage. The next thing the author want to ask is that whether these exosomes can dose-dependently regulate the immune response. And so the author will culture these exosomes that derive from 48-hour hypoxia or 24-hour hypoxia with different dose. And they use RT-qPCR. They examine the inflammatory they examine the expression of the inflammatory molecules inside this macrophage. And figure C is a 24-hour group, and as we can see that in this group, first of all, we can see exosome can actually dose dependently increase the secretion of these markers, no matter it's coming from the control or the hypoxia group. However, though, in 24 hours, the compare between the control and the hypoxia group, there is no significant change in this marker expression. In the 48 hours, we can also see that there is a increase when macrophage engulf these TEC derived exosome. However, unlike the 24-hour group, in the 48-hour group, the hypoxia exosome from the hypoxia group can significantly increase these markers compared with the control group. And here we can see that at the dose of 15 and 30 nanogram per milliliter, milliliter here, L is lost. Okay. At 15 and 30 nanogram milliliter, the inflammatory molecules are significantly increased. So the author decided that all subsequent experiments will be performed using 15 nanogram per milliliters of exosome. Okay. Wow, we are like two out of 3 of this scientific report. Thank God. So that is dose dependent. Now we want to know whether there is a time dependent. And so using 15 nanogram per milliliter of TEC derived exosome in the 48-hour hypoxia group, and the other trick, we can see that at different time point, as the time gradually as over time, there's a increase. How do I say that? Over the time, there is a significant increase of the inflammatory markers in the hypoxia group. You can see here. Yeah, we can just call it hypoxia group. It's just so complicated. The name is just so complicated. The saying is every time, it's just hurting my throat. Okay. Also, we can see that over the time, phospho P65 also significant increase, and this increase is significant in the hypoxia group. So these findings indicate that exosome released from the hypoxic TEC in the 48-hour groups can promote macrophage activation. Okay. So in the smaller scale, we want to know that whether the mitochondria inside the exosome actually interact with macrophage during our issue. Okay, so these mitochondria mimic are conjugate with CY3, which will deliver a red color, and they are then transfected into TECs. TECs containing these CY3 mitochondria mimic will then undergo hypoxia for 48 hours. Then the exosomes are purified using differential centrifugation and applied on macrophage. As a result, in figure B, in the immunohistochemically, wait, it's not immunohistochemical, right? Immuno or whatever. Let's forget about it now. So in figure B, we can see that there is an increase in the internalization of the red dot, which is the mitochondria inside the mice macrophage. So to test out the effect of these exomitochondria, the order perform gain of function and loss of function. They either inhibit these mitochondria or overexpress it, and as a result, using RT-qPCR, we can see that in terms of inflammatory markers, these old guys, we can see that inhibition of mitochondria will decrease the expression of the inflammatory markers compared to the negative control, and overexpression of mitochondria will increase the level of these markers. So the next thing the author wants to ask is that what is the potential target of mitochondria in this manner? So they use the computational method to predict this target. They use target scan RDB, Miranda, Diana, Tarbase, and Victor. So overlap analysis revealed that the partial target is A20. It is a negative regulator of NF-kappa B. And so, having found out the potential target, the author will test whether it is actually regulated by mitochondria. So we can see that in terms of Western blot, if the mice is exposed to this hypoxia mitochondria over hypoxia exosome over time, the expression of A20 will decrease, as you can see in the quantized graph here. And to find out whether there is a direct interaction between mitochondria and A20, they use a luciferase assay to address this question. And as we can see that compared to the pre-UTR negative control mitochondria, the pre-UTR mitochondria can significantly decrease the activity of the reporter, and the mutation of the pre-UTR of the A20 pre-UTR can abolish this effect. So here they confirm that mitochondria can directly interact with A20, and now we're gonna see that in terms of Western blot, using gain of function and loss of function and gain of function, we can confirm that in loss of function, in gain of function, A20 expression level are inhibited, and in loss of function, the level of A20 were increased in these mice macrophage. So in short, these data suggest that these TEC derived exomitochondria, okay, is hypoxic. this hypoxic TEC derived exo, add the word hypoxic TEC derived exomitobriae so long activate macrophage via regulation of A20. Dang, yeah. Okay, last. We finally moved to here. Thank God. Thank God. Almost spent about an hour. Okay, so let's just walk through the end of this. Okay, so that's we have already finished the in vitro model. Now is the in vivo model. The same thing, we will look at the bigger scale, whether there is an interaction, whether there is an effect of TEC derived exosome on the progression of TI in mice, and whether the mitochondria from the exosome can have is actually the one that exerting this effect. Okay, so in terms of the exosome, the bigger scale, you can see that addressing the administration of this hypoxic exosome will damage the kidney tissue more compared to the control exosome. Investigating the whether these exosomes are and mitochondria internalized in macrophage, we can see that the green CD68 is macrophage, it stands for macrophage exosome, and mitochondria are sent with the green color, and DAPI is the cell nucleus. As we can see that compared with the control exosome group, in the hypoxia exosome group, we can see there's a significant increase in the exosome inside CD68 macrophage, and also there's a increase in the internalization mitochondria in the macrophage in the hypoxia exosome group. Okay. Also, in RT-qPCR, we confirm that the treatment of these hypoxic exosome will increase the inflammatory molecules phospho P65 and decrease A20 level. Okay, so finally in the the smaller scale, the exomitochondria effect on mice. Well, actually, is it a similar pattern? We can see that to check whether mitochondria is actually like the main character here, they use loss of function experiment, so they will inhibit mitochondria in this hypoxic TEC derived exosome and see whether this can improve the repair of the kidney tissue. As we can see here, compared to the exosome group, inhibition of mitochondria can actually improve it, and there is a decrease in the infiltration of macrophage. Also, the inflammatory molecules, the level decrease, as well as phospho P65, and there's an increase in A20. So, to the conclusion of our study here, TEC, alpha VC, in hypoxia, HF1 alpha will increase in the TEC, and this TEC will respond by secreting mitochondria enriched exosome. These exosomes are received by macrophage. Inside macrophage, mitochondria will inhibit A20, which is a negative regulator of NF-kappa B, and in this manner, it will reprogram macrophage into a pro-inflammatory state. As a final consequence, TI will be formed, and this is and this is the findings of this study. And by learning about this fact provide a potential therapeutic approach that maybe from the blockage of the exosome in the early stage, we can preventing TI formation, or we can ameliorate the damage that our kidney take to improve the damage that our kidney take. And that is all I'm about to say today. Thank you for everyone who's sticking with me to the end. It's like about an hour. Yeah, it's just exactly an hour. I started 8. Yeah, I started 7, about 7:20, and finished and about now it's 8:20. Yeah, Q&A. No Q&A. Fucked up. Thank you for your attention. 报完了1个小时，但真的好干哦，太长了啦！为什么我要挑这篇 paper？ 为什么？真的是怀疑人生欸。不对，我应该要想出一些计谋。我先放一个很香的图，然后我来讲这件事情。我要思考一下。好，对，我应该要把前面那些东西都讲一遍，然后讲好之后，我要跟大家说 OK， 接下来我就要快速冲，不然就他妈的真的就是这个真是很泥淖有点掉进去一个坑里面，感觉。要不要放这个？放这个好了。稍微整理一下我的思绪。嘴巴好干哦。放这个应该比较香。OK。对，应该要那样。对，所以我应该有几个部分回到我们的东西，确定一下。Introduction 不用说，一定要说清楚的。觉得从这里，其实从这些图啊，我讲了一次之后，应该就不用再重复了。就是说，下次我看到这种图的时候，就直接讲它的现象代表什么，或者就是说有类似的现象。对，这前面就这样讲了，那后面这该从什么？到这里还不算卡，到这里其实都还好，到这里开始就是真的是有点泥淖了。这里开始真的他们研究。这些地方啊，像这张图带过，就是 RTQP 这种的。干那个标题这么大，其实有点卡位。好，我先放着。应该说 24. So I shouldn't say that 24 group, hypoxia 24 group, or control 24 group. Yeah, just don't say something like TEC derived exosome from the hypoxia 24 group. Just a fucking long. Maybe I just say something like exosome from hypoxia 24-hour group or exosome from hypoxia 48-hour group, hypo 48 group. Yeah. If they don't get it, they might just need to ask. That will be better, I guess. So exo hypo 24-hour group. Yeah, in exo hypo 24-hour group. Yeah. Just make things short. Okay. Or just about the logic. Yeah. Okay. Okay. Thanks everyone for sticking with me and the stream end here. 那谢谢大家今天早上来陪我这个有点胡闹的直播。我今天晚上搞不好还直播一次哦，就是应该也会再报一次一样的模式。然后对，然后明天上阵，然后今天晚上我应该会再报一次这样 ，OK？ 那谢谢大家，我们直播就到这边，大家啵啵。